Hello everyone, my name is João Vitorino. I'm a PhD student at the Computational Biophysics Lab, Miguel's lab, from the University of Lisbon Faculty of Sciences. And I'm going to tell you a bit about our work on protein aggregation. So, not all aggregation is bad, just like not all amyloids are bad. However, it is a problem when proteins that shouldn't aggregate do. There is a case for Alzheimer's disease, Parkinson's disease, and dialysis-related amyloidosis. In this last case, the accumulation and aggregation of beta-2 microglobulin will lead to a number of problems and conditions in patients undergoing long-term hemodialysis, since this procedure is not able to clear it from the plasma. That will ultimately result in organ failure and even death. Beta-2M generally in its wild type form does not aggregate, but under physiological conditions and under physiological concentrations. But there is a particular mutant, the D76N, that is found in some people that does aggregate. This mutant tends to populate a particular intermediate state called I2, where both the N and C termini detach from the core of the protein, exposing the more hydrophobic residues, and that leads to a higher propensity towards aggregation. This was actually studied as well in collaboration with another group at our faculty, the group of Professor Patrícia Faísca. And more recently, we've been focused on studying the earlier phases of protein aggregation using this D76N mutant as a model. In 2019, these structures were simulated using constant pH MD, and the resulting ensemble was used to form configurations of dimers of this mutant. We used Monte Carlo ensemble docking methods that Professor Patrícia Faísca had previously created to generate thousands of these dimer configurations. And using a stability cutoff, we selected 212 dimers which were plausible. And then we, to assess their stability, went on to do an interfacial or structural relaxation of these dimers with short MD simulations. So there are many metrics we can follow during MD simulations. However, maybe the more direct one would be actually to calculate their binding free energies. So how can we calculate binding free energies? It isn't trivial to get directly this delta G of binding in solvent, but of course, there are many methods that we can use, and that in this conference have been mentioned and overviewed over and over again. So I'll just briefly talk about a few ones. So starting from docking, docking has the problem of having very weak sampling. And while it has lower computational cost and lower complexity, it also suffers from lower accuracy. On the other end of the spectrum, we have alchemical perturbation methods that, while they provide extensive sampling, they have other types of problems, such as, well, the computational cost, of course, and having unphysical intermediate states. So we ended up choosing one of the so-called endpoint methods, molecular mechanics, Poisson-Boltzmann surface area methods, which I believe Robert overviewed a bit, or Christopher. Yeah. And we actually made a new implementation for it, which works in Gromos and Gromax, where we run our MD simulations. So when we use PBSM methods, we are attempting to use a thermodynamic cycle such as this one to get to this delta G of binding in solvent. So we approximate the enthalpy to delta G of binding in vacuum plus a solvation term. Here is the general outline of an PBSA calculation. So we have a vacuum term, like I said, which we can get through molecular mechanics. I'll show you a bit in a bit the equations, and the solvation term, which comprises a polar and a non-polar term. The polar term we can get, of course, calculating using a continuum electrostatics method to solve the Poisson-Boltzmann equation, and the non-polar part we can get using a surface area model. So, as for the molecular mechanics part, we can calculate this part using the same types of calculations we do when we run MD simulations. So we have our bonding interactions and our non-bonding interactions. However, typically when we use PBSA methods, we also use an approach called single trajectory, which by grabbing the conformation of the complex, the ligand and the protein from the same frame from a single trajectory, this allows for the bonded terms of these structures, the bounded and unbound structures, to have the same bonded terms, so they cancel out. As for the entropy, calculating the entropy is very heavy, and it doesn't improve the correlations with experimental energies very much. So although it's not realistic to say they cancel out, we're only basically ignoring the extra flexibility the unbound species might have over the bound complex. And this, for protein aggregation, we hope it isn't very substantial, of course. And we're getting the flexibility somewhat, although not very, accounted for in the MD sampling space. So as for the Poisson-Boltzmann equation, like I said, we can use the continuum electrostatic calculations, where we define a solute with the dielectric constants, typically very low, and a dielectric constant for solvents, typically water is 78 to 80, and we basically solve the Poisson-Boltzmann equation for all the points in the grid, and that way we can get the delta G of solvation, the polar contribution. Finally, we have surface area. We can use the surface area model where we use a molecular probe, typically water with a radius of 1.4 angstrom, and Lee-Richards algorithm in this case to calculate the surface area, and use a model where we can input this value and extract directly delta G non-polar contribution. So this was the outline of our binding free of our MMPBSA implementation, which is called PyBindy. PyBindy is available on our GitHub page along with some other very nice tools. So check it out. And okay, using PyBindy, we found that our Monte Carlo instable docking method provided mostly decent starting configuration, since after the MD relaxation, although their binding interfaces may vary, all the dimers possessed negative binding energies. And we also determined that the binding is in this ensemble is mainly driven by non-polar interactions. With this, we were also able to assess the binding interfaces of the dimers, and in particular, which residues are more likely to populate the interfaces of the dimers with the 10% best and 10% worst binding energies. And we found that the residues that contribute to the 10% best binding energies are typically the ones in the N and C termini, and the ones in the DE and BC loops. This was something we were very happy about, since it's something that matched the literature as well. So now we had too many configurations, so much so that some of them were probably not unique. So what we did was we used the clustering protocol to find unique binding modes, and which we ranked according to their stability. We saw that for the best 10 binding modes, the interfacial area and the binding free energies tended to correlate somewhat, and a bigger interfacial area tends to translate to a lower binding energy, although not always. This, of course, depends on the residues that are on the interface and their complementarity. These interfacial areas from the initial MCED gas, we're talking about more than 60% conservation, although there was one outlier, the binding mode 4, which changed almost completely its binding interface. This was probably a dimer configuration which found a more stable minimum through MD trajectory. Okay. So now we had the list of unique binding modes, and we ran 100 nanoseconds of MD simulations. But is 100 nanoseconds really enough to know if they're stable over time? We decided to see how they would behave in longer time periods, if they remain stable, if they dissociated, and if ultimately growth mode based on these interfaces would be at least viable. So we picked 10 of the highest affinity binding modes, 3 with average affinity, and 2 with the lowest affinity. And we ran much longer simulations, triplicates of 500 nanoseconds each. And we once more calculated binding free energies using PyBinding. What we saw was that there was, once again, a correlation between the interfacial area and the binding energies. And we also saw that the binding energies tended to stay almost the same, with a slight tendency even to further decrease. Now, we had a list of dimers whose interfaces are candidates for aggregation. But let's assume that they're all sufficiently stable to withstand growth. We decided to analyze how these binding modes could result in polymerization, and if it would be at least geometrically possible to replicate them in space. So in the early phases of beta 2M aggregation, the monomers tend to stay well, spherical a lot. So let's think about our monomers as simple spheres. Now, monomer B has a given spatial orientation in regards to monomer A. And if we were to add a third monomer, we would need to find where in monomer B would this monomer connect to A using the same interface between A and B, which would be right about here. And of course, the same for subsequent units. This expansion can be characterized by a polymerization angle, which is basically the angle between the vectors from the center to the interface for each two monomers, and polymerization dihedral, which accounts for the 3D torsions that might occur between monomers. Okay, applying this polymerization model to our binding modes, we saw that the growth potential could be either unlimited, where the binding modes allowed for the continuous addition of new subunits, and that led to an unlimited potential to grow, or a limited growth, which was based on angles and dihedrals that produce an overlap of when we try to add another monomer to the chain. So eventually that growth mode would be limited. Of course, this works under the assumption that each binding mode can support this type of growth, which would not always be the case, but we're assessing the binding interfaces, so we have to think about it like this. So we apply this logic to a representative structure of each of our binding modes for all the replicates of each of our binding modes, and see how they grew over time. Of course, we could do this for all the the frames of our simulation, but it's 22.5 microseconds, and having to add all these monomers to all the structures at each point in time, and then having to inspect it visually would be, well, very heavy. Okay, so now we had a way to calculate polymerization angles, polymerization dihedrals, and then by adding the monomers to a chain using the same polymerization angles and dihedrals and inspecting it, we could assess growth potential that way. But of course, this would be unfeasible to see all the conformations in our trajectories. So we started thinking about a way to directly grab these polymerization angles and dihedrals and assess growth potential. So what we did was, using our sphere model, and for the sake of simplicity, we constructed a plot which can visually help us identify if a growth mode will be limited or unlimited. And that, of course, is much easier than, well, the other option. So we can see that for angles that are very low, our binding modes will be very limited. It would produce too much overlap, like we can see here in this binding mode 1. On the other hand, for binding modes that have very wide dihedrals close to 180 degrees and angles over 90 degrees, which produces this kind of linear growth, which has an unlimited potential. Similarly, but with dihedrals very low, we have this behavior which produces funny helical growth, since while the angle is high, the dihedral is not. This pipeline we developed is also completely general, so it cannot only be applied to beta 2M, but other proteins as well. So now we have a protocol which allows us to predict the potential to grow of any potential structure. Finally, I wish to thank FCT, Fundação para a Ciência e Tecnologia, for the grants and projects which finance this work. My supervisor, Dr. Miguel Masqueira, of course, Professor Patrícia Fisca, our close collaborator on this work, and my whole lab and my friends, which are right there, and the organizing committee of this conference, and all of you, of course, for your attention.
Okay. Any questions? At the beginning of your talk, you mentioned that two beta strands are detached when the protein aggregates, right? So what kind of experimental evidence do you have? That's one question. Another question is, for this kind of filament, probably cryo-EM can be directly applicable, right? So compared to such potential cryo-EM studies, what could be the advantage of your computational work?
Okay, that is a tough question since I'm mostly a computational biochemist, but I'll try to answer. We have experimental evidence that those mutants have detached arms. It's not us as in our group, but there is in the literature evidence that these structures populate that intermediate state, which unfolds its arms. Now, as for the cryo-EM, I don't have a lot of experience, I will be honest, with cryo-EM, so I don't know if I would be able to answer appropriately. Sorry.
Thanks. Yeah, great presentation. And the visuals are pretty awesome. I have two questions. The first one, with the non-polar part you're doing with the SASA model, right?
Okay.
You're directly doing the whole non-polar term, or are you
The whole non-polar term.
okay?
There are several models we can use. That one is only dependent on the surface.
I agree.
the
No, I
assessment
think
surface.
that you can further decompose it into the cavity and a
Yeah,
dispersion term, and
this is an
actually, and
approximation.
those two by themselves correlate better with SASA. They both correlate with SASA, but with different constants.
Yeah, but they are also tougher to calculate.
No. You use exact same equation.
Oh, okay.
You just so you would have two SASA models, one for the dispersion term, and one for the cavity term.
Okay.
And it's usually, I don't know if more accurate, but the correlation is better.
All right. What we saw when we were implementing this and writing this implementation was that the SASA model, only dependent on SASA, also correlated very well with the experimental values. So we thought that this one was maybe faster, but we actually didn't calculate all the ways that we could. So that's maybe
it's
a way to improve.
I've always wondered, because usually looking at MMPBSA papers, they show your equation, but there's other sort of stream of papers where they show that if you decompose them, you can get a little bit more accurate. But I guess it's in the end you're still fiddling around with the parameter there, so you can.
Yeah.
The second question is related with what Junji was mentioning. Because at some point you said that the entropic term you're not considering, essentially not considering any conformational changes during the binding, right? So in your initial state or isolated state, are the strands already opened up?
I don't think I understand the question.
In your thermodynamic cycle,
Mm-hmm.
you have the isolated state, right? And then you have a bound state.
Yeah.
In the isolated state, are the strands already opened, or are they closed?
They are the same conformations that we have in complex.
That was my question.
Yeah.
Okay, thank you.